Hello, and welcome back to Beyond Networks, the evolution of living systems. We're slowly but surely getting into the evolutionary part of this lecture. And in this module in particular, we're going to be mainly focused on the question of how do you get novelty and innovation in evolution. This is a really important problem that was already recognized by Charles Darwin in his Origin of Species. He says, characters may have originated from quite secondary causes, independently from natural selection. And very famously, later on, Hugo de Vries, one of the mutationist geneticists, says natural selection may explain the survival of the fittest, but it cannot explain the arrival of the fittest. The problem, of course, is that selection acts on variation that is already there. In order to truly understand where evolutionary innovation comes from, you need to understand the sources of this variation. And so this is basically the causal completeness principle again, that you need to understand development to understand evolution, because evolution is working by changing developmental processes. I'm going to use a slightly different phrasing here by James Grissomer from a wonderful book chapter from 2006 that I highly recommend. And in that book chapter, he writes, the evolution of new traits, and we're going to quickly divert into that, new levels of organization requires the evolution of new ontogenetic processes, not only the evolution of adaptation. So especially for innovations, you need to understand development, and you cannot separate it from other processes that are going on in evolution, like it's often done with population level evolutionary genetics. So what does he mean here, new levels of organization? We're linking right back to Bill Wimsatt's perspectivism and his theory of levels of organization. Not by chance, because James is a student of Bill Wimsatt. And so let's divert quickly into this type of novelty. So there are novel traits, novel functions. We're going to come back to that. But there are sometimes in evolution entirely new levels of organization. And this is the topic of a wonderful little book called The Major Transitions in Evolution by John Maynard Smith and Urs Satmari, which was written in 1995, updated by Urs in 2015. I'm going to show you here the 7 major transitions that happen in evolution according to this revised version of the theory. So the idea here is that sometimes, very rarely, but sometimes, something completely new comes into the world. And of course, the first such transition is a protocell. Genes, chromosomes, some sort of mechanism of inheritance in a compartment has to be localized. We were talking about this when we were talking about autopoietic systems. They have to show autopoiesis, but also be compartmentalized. This is the first major transition in evolution. The next step is prokaryotic cell, like we know it today, with a genetic code. So we went from RNA molecules working as enzymes and molecules carriers of inheritance to DNA, proteins, bacterial sex, the exchange of plasmids. And then, of course, the next step are eukaryotic cells that not only come with the invention of meiotic sex, but also endosymbiosis. Mitochondria are bacteria that were incorporated into the cell. And so there's a separate step here for the plastid, acquisition of plastids in plants as well. But that's very similar to step number 3. Okay, so these are endosymbiosis events, where instead of divergence in evolution, you get something coming together, merging and synergizing, instead of competing with each other. Those are very interesting steps. You have the big step of the evolution of multicellularity, which sort of involves 2 aspects. One is cell differentiation, division of labor, especially between the soma and the germline. So the majority of the cells in the body now sacrifice themselves for a few in the germline to propagate. And then you have eusocial animal societies, such as anthill, social insects, for example, where you have to have mechanisms that control conflict between individuals, and you get this idea of the superorganism. And one eusocial species, of course, a very special one, that's ourselves, the human species. And we have made one major step, again, in that we have evolved language. And so we get an entire new level of evolution, cultural evolution, and cooperation between non-related individuals, which is, as far as we know, unique to human beings at a major scale. So these are 7 major steps where you get novelty that isn't just a new trait. It is an entirely new way of reproducing. New systems of inheritance. We'll revisit this in the next module. But basically, each one of those steps, very special steps in evolution, has 3 major common features. Okay, so one is a transition from independent reproducers to higher level units. Multicellularity. Each cell used to divide itself, and now the somatic cells sort of sacrifice themselves. Okay, and the new individual is the multicellular individual. You get a division of labor, soma versus germline, different differentiated types of cells. You get a novel inheritance system. Cultural evolution is the most striking example, where you get transmission of information through what some people call memes, and they say, oh, these are replicators like genes. We're going to revisit that as well in the next module, that sort of view. Okay, so these 3 sort of features, a transition of the unit of selection, really, to a higher level of organization, a diversification, a division of labor, and new sort of inheritance systems are typical for each one of those 7 major steps. Okay, so these are rare steps, but they're huge. Each one of them has a huge influence on evolution. And also, they're very special because they come with a new set of rules. With it's a new game that's being played every time something like this happens, especially at the origin of life. When life comes into being, all the rules, the whole organization of the living system comes into place for the first time in the universe. So sort of the things that can possibly happen in the universe have suddenly expanded massively. And so we're going to talk about this type. These are emergent sort of events. And the new rules are emergent properties of a system in a very strong way. So we're sort of in a very strong sense. So we're going to talk about this a little bit more. But for the moment, I want to sort of come back from, just keep this in mind. This is sort of a major sort of way of evolutionary innovation. It's huge compared to just sort of the appearance of a new trait, a character trait, that we're going to focus on right now. So what I want to do for the rest of this lecture is I want to introduce 2 ways of dealing with innovation in traits. Okay, so let's come back to James here. He's saying we need to account for these innovations, big at the level of organization, but also small, just a new trait. We need an account of how heredity and development intertwine to co-produce conditions of evolvability. Okay, so we will not understand these events if we do not understand the mechanisms that produce either a new level of organization or this new trait. If we don't understand the source, only knowing the consequence rules is not going to help us understand this. There's a lot of debate whether, innovation is really qualitatively different from adaptation. But I would tend to side with those that say, yes, this is different. Okay, so to look at this, we need an explicit process perspective on evolution, right? And I was showing you this slide the last time. So we need to sort of fully realize that evolution and ontogenesis and phylogenesis and ontogenesis are the same process at different time scales. And only by understanding these ontogenetic processes that create a closed life cycle can we understand evolutionary dynamics. Okay, and so I was using this very useful separation between the sources, the circumstances that produce heritable fitness variation, and the consequences that describe how heritable variation in fitness, once it exists, leads to change. These two aspects of evolution need to go together for a full understanding of what's going on. So the way last time that I said evo-devo in particular, evolutionary developmental biology, is tackling this, is by providing a type of explanation for evolution that goes beyond the division that Meyer made between proximate and ultimate causes. And these are lineage explanations, a term proposed by philosopher of science Brett Calcott. And as I told you last time, the way it works is that you have sort of a production rule here, which is described by one of those dynamic mechanisms that we were introducing in the first part of the course. And you want to explain how this changed through evolutionary time. Okay, so here, this abstract but simple example uses letters to form 5-letter words of the English language. So that's your production rule. It has to make sense as a word at the end. So here you have scale, and then you have a continuity rule which says you can only change one letter at a time. So mutations only affect one letter. And you want to get from scale to plume, but you want to do this through intermediates that also are words of the English language that fulfill a certain function, okay? And you can easily do that with any sort of 4 or 5-letter words combination in English, surprisingly. You will find a pathway between the two that goes exclusively through functional states. Okay, to make this more concrete, I introduced this already last time, but here you have an example, and this is the evolution of feathers from scales in reptiles and dinosaurs, such as birds. So feathers are these intricate structure with a stem called a rachis and barbs, okay, that sort of interlock with barbs and barbules. It's kind of a fractal structure. It's beautiful, much more complex than the initial scale. Okay, so how did this complex structure originate from a scale? What changed, obviously, is the way that the feather is grown. And it turns out, and this is wonderful work by David Prum, that you can show that by making small changes to the sort of pore, the shape of the invagination here that produces the scale or the feather, by changing the shape of that, you can get all these different structures, and you can get this refined, this amazingly complex structure of a feather. By relatively simple changes in the developmental process, you get this sort of emergent complexity of the phenotypic trait. Okay, and now this is considered as a true evolutionary novelty. Feathers have a different function than scales. They initially, and they have a changing function in evolutionary history, initially they were probably for thermoregulation. And then later on, of course, with birds, they were important for the evolution of wings for flying. Okay, so this is an example, remember, of exaptation, where you have a trait that is evolved for one function, thermoregulation, being sort of co-opted into another function. And by the way, the function of flying is a novel function. We'll come back to that in just a second. Okay, so this is a very interesting example, and we're going to use it quite frequently in this lecture. So we want to deal with such an event. How can you get the evolution of feathers? Does it need anything more than just natural selection? How is a feather produced in the first place? How is the innovation produced, generated from its sources? And so we could say, okay, we have two different phenomena to explain. One is just adaptation of an existing trait, and one is the source of the novelty. There's a beautiful paper by Gerhard Müller and Günther Wagner from 1991 that does a very careful conceptual analysis of this term novelty. And it makes a few really useful distinctions. So I'm going to introduce them here. One distinction that they make is they separate innovation from novelty. they call an evolutionary innovation the acquisition of a new function. So birds taking to the air. So there is now selective pressure for this new function of flying. And then it goes backwards with penguins, right? So the feathers and the wings of penguins are for swimming and thermoregulation again. So it can go back and forth. So the acquisition or the sort of emergence of a new function is called an innovation. This will be the main topic of next lecture. Today we're going to focus on structural novelties. Okay, and that's what Müller and Wagner call a novelty in the strict sense. It is a structure that is neither homologous to any structure in the ancestral species, nor serially homologous to any other structure of the same organism. Okay, very briefly, what is homology here? Homology is central to this definition. Homology was defined by Richard Owen, it's a beautiful picture of him here, as the same organ in different animals under every variety of form and function. It's a great definition because it's perfectly accurate and also perfectly useless in practice. Because under every variety of form and function, what does that mean? The same organ, what does that mean? The same organ nowadays, of course, is interpreted as a structure that descends from the same ancestor. And often we take sort of the structural context of an animal into account. So the developmental origin of an organ gives us a lot of hints towards whether an organ is actually homologous or not. Okay, so a novelty is an organ that is not homologous to any structure in the ancestor. It's something new. And it's also not serially homologous to any other structure of the same organism. Serial homology is sort of the homology between your forelimbs, your arms, and your hind limbs, your legs. They're both homologous structures, but they're variations of a theme that happen in your own body. And so serial homologs happen, for example, insect forewings, hind wings, and so on and so forth. So a truly novel structure is neither homologous to any structure in the ancestor nor to any other structure in the same animal. And so since 1991, the paths of Gerrit Müller and Günther Wagner have diverged. We're first going to follow Gerrit for a little bit and then look at Günther's theory. Okay, so Müller and Newman in 2005 are writing mechanisms by which a new homologue is introduced into an existing character assembly are rooted in the emergent effects of interacting levels of biological organization, molecules, cells, tissues that are mobilized during evolutionary change. Okay, so what they're saying here, I think, is that this is not just a genetic process, okay? So they focus in a holistic manner on sort of all kinds of levels of organization in an organism. Okay, what do they mean by mobilized during evolutionary change? I don't know quite yet. So let's explore this for very briefly. Müller calls his theory epigenetic innovation theory because it goes beyond the level of genetic evolution. And what he does is he distinguishes between initiating and realizing conditions for novelty. Basically, he retains Meyer's distinction between ultimate and proximate sort of causes or explanations. So initiating conditions explain the evolutionary origin of a trait. Okay, so you can think of them, for example, it could be a functional shift or a functional decoupling or an exaptation. Again, bird wings, feathers evolved for thermal regulation, the wings themselves probably for gliding or something like that, and then they were exapted into the function, the novel function, the innovation of flying. Okay, so this is a shift of selective pressures that you can only explain through an interplay between the structure that's being selected and selection. Okay, then there are internal mutation-based mechanisms, duplications, that, for example, gene duplications that can give you the sort of conditions under which you can evolve novelties, okay? We're going to come back to that, not just in this lecture, but also later on when I talk about network evolution. And so you can have environmental induction, phenotypic plasticity, and phenotypic accommodation. So you can have a phenotype that is already very sort of flexible, and it can adapt to environmental conditions through its intrinsic dispositions. Okay, so you can actually evolve and fix traits that are plastic. That's no problem. But here they're saying this tendency to be plastic can be used to innovate. A plastic change goes through a population directly. Every individual will go through it. So it's much more efficient in many ways than sort of selecting for a rare mutant. And they do admit that natural selection will work together with all these different circumstances. Okay, so you have all these sort of conditions are required for the evolution of the novelty, but then you need some sort of internal factors as well, which they call realizing conditions, proximate factors. And I have to say, Müller and Newman remain rather vague here. They say that there's some sort of threshold effects and some forms of self-organization. Especially Stuart Newman has elaborated a very sort of careful theory about what he calls dynamic patterning modules that explains what he means by self-organization. But in this sort of general sense, it's rather broad terms, okay? So how is this supposed to be informative? Here it is. So epigenetic innovation theory makes a few statements that you can summarize like this. The first is that realizing conditions, so the ultimate conditions for innovation, the proximate conditions for innovation are embodied in developmental systems. Okay, so they're basically saying, just like the Vries, you have to know development or the source of these conditions that are internal. They're not natural selection. Self-organization, feedback regulation leading to threshold effects, and environmental dependence, phenotypic plasticity, et cetera, et cetera. They introduced this term morphogenetic templates for phenotypic evolution. These are sort of generic processes underlying development. They're typical processes that happen over and over again. So, for example, excitable cell aggregates and their emergent properties. So we were talking about this when we were talking about process structuralism. This is going to come back in the next module. But here's the idea. There are certain typical forms that happen much more often than others because morphogenetic fields have a certain structure. There are constraints on those fields. There are not endless forms most beautiful, like Darwin said, but only a limited number of possibilities. Remember Tome's theory of the different catastrophes and how they were actually really limited in number. So you could think of these morphogenetic templates a little bit, very vaguely, like Tome's catastrophes. Okay, the next point, the developmental systems are complex, they're non-linear, they react in unexpected ways to changes in parameters, initial boundary conditions. So it's not just a gradual, predictable change. And so this unpredictable change can be the source of innovation. Robustness, it's very important. We're going to revisit this several times in the course still. So parameter changes are usually buffered until suddenly a threshold effect occurs. And this means that evolutionary change must happen through discontinuous phenotypic outcomes. I was making a few lectures back this argument based on dynamical systems theory. You can make this argument from first principles. This has to be the case. So you just can't evolve by continuous change only. So what you need to do is you need to think, how does this continuous sudden change get incorporated into evolution? You can't just deny its existence, okay? Because it's impossible to evolve otherwise. So we can think of the loss of traits, trait fusion, or the de novo origination of new kernels for new traits. The basic point of all of this is innovations are only elaborated upon by natural selection, but the actual source, if you want to understand that, you need to understand the developmental system that produces the variation. Genetic evolution consolidates rather than invents innovation through morphogenetic templates. Now, this is just the Vries, right? Okay, so this is very complicated and it's very nice, but it uses more words than the Vries, but is it really more specific? I don't know. What do you think? I don't think so. There is no progress here compared to the Vries at all, because there are no specific mechanisms, and it's all very sort of generic, right? So we're not making real progress here, I think. And there's a different and really big problem that I'll come back to at the end of the lecture with this theory. But this is sort of the state of theorizing, especially in evo-devo, okay? So you have a verbally formulated theory about a bunch of interesting concepts, but unfortunately it remains at this sort of stage where it sounds interesting, but it's not further defined what emergence is in this context, for example, what these nebulous self-organizing threshold processes are. It's missing, okay? That's a problem. So let's get back from this sort of very broad theory to Günther Wagner's idea of innovation. And here is a very different sort of approach, okay? Wagner writes, the origin of an evolutionary novelty is the evolutionary process through which a novel character identity arises. I'll come back to that in a second. In other words, an evolutionary novelty originates when a part of the body acquires individuality and quasi-independence. This is really interesting. Okay, something becomes individualized. Okay, only like this can it become non-homologous to another trait. It has to become a trait first. If it's not independent, if it cannot vary independently, it is not even a trait. So it cannot be compared to other traits. We cannot even know if it's homologous or not. And Wagner uses the example of the human chin, which is a funny structure because it cannot really be selected for because it originates from two different morphogenetic fields that interact in the human face during embryogenesis. Okay, and so this is not a trait because it cannot vary independently, and so it cannot be an innovation. It cannot be selected for independently. So what does he mean by character identity? So Wagner makes this very interesting and powerful distinction between character identity and character state. So let's take the examples of insect wings. The prototypical insect has two pairs of wings, a pair of forewings and a pair of hindwings. And in most insects, both of them are used for flying, like in this beautiful butterfly here. But some groups of insects, like the Diptera, mosquitoes and the flies, have altered their hindwings, in this example, are reduced to these balance organs called halteres here. The arrow points to them, and only the forewings are used for flying. While others, like beetles, Coleoptera, have modified their forewings to be covers, protecting the actual flying wings, which are now the hind wings here. So in these cases, character identity is being a forewing or a hind wing. So it's broad sense homology based on continuity of lineages. Okay, so the forewing here is the same, has the same, descends from the same ancestor as the forewing here, but their function has drastically changed. So this is reflected in the character state. Characters vary in size, shape, and color. So whether something is a flying wing, a haltere, or an elytron, that is a matter of its state, not its identity. And the best way to compare homologies is at the level of character identity, not the state. Okay, so it's not that the flying wings are homologous to each other, but the forewings and the hind wings are in this case. So Günther now goes a very different way from Gerd Müller. He focuses in on the genetic level completely. So gene networks are all the rage. So he says, okay, so character identity needs to have a genetic source. Okay, so if you want to understand the source laws, you need to go to the genetic level. And his theory is that developmental processes are generally structured like this. There's some sort of general positional signals that specify a sort of intermediate network of transcriptional regulators that he calls chin. character identity network. Notice the joke. The chin is actually not a character with any identity. Okay, and these chin networks, in turn, they regulate the realizer genes that make a wing, haltear, a flying wing, or an elytron. The structural genes, okay, which are responsible, which he calls realizer genes, which are responsible for character states. Sort of similar to Eric Davidson, okay? And so these chins is what you need to study to understand where characters originate. So he diversifies his concepts a little more. And for that, we need to sort of understand. So he wants to look at the origin. Where do these novel character traits come from? He says there's two different ways. And one of them has something to do with a rather drastic change in the state, but not the identity of a character. Take, for example, the fin to limb transition. When some kind of lobe fin fish came on land. Here is Tiktaalik, famous fossil as the fish that can do push-ups, intermediate form, and here's a terrestrial tetrapod. So there's no doubt that a limb of a tetrapod is homologous to the fin, the paired fins of fishes. But it is so drastically different that it's barely recognizable. And Wagner calls this a variational modality, okay? So if you have sort of the same character identity, but very drastic and irreversible changes, that is a type of novelty. So that the vertebrate limb, tetrapod limb, nobody would doubt that this is an innovation. It came with a new function, that's walking on land. Okay, so that's the innovation. The novelty is this structure, but it's based on the old fin. So how do we treat this? Okay, the important thing to know about modalities is, as I said already, the transition from one modality to another involves developmental changes that are major, probably irreversible. And so non-overlapping transitions among character states within each mode are much more frequent than between modalities. They're rare. So a new modality is very rare, just like a new trait. It is expected that variational modalities differ in their developmental functional organization and thus reflect mechanistically relevant differences among sets of character states. So there must be drastic difference in the network underlying the trait. To summarize, so there's two types of innovations. Type one is this novelty in a strict sense, okay? So that reflects the origination of a truly new character identity. Okay, something has become individuated, has become a new trait with a new chin underlying it in this theory. And type two, the origination of a new variational modality, a rare irreversible change of an existing chin. Feathers are another example. A feather is a different modality from the scale. It's not an entirely new part of the body, but it's absolutely not recognizable anymore. In both cases, and that's important, you need very drastic changes to the regulatory network to get the evolutionary transition. So in this theory, you can identify different developmental and also genetic mechanisms for novelty. And the idea is to look at them and see if they're the same or different from those that we use to explain adaptations. This is described in a lot of detail. I don't have the time, unfortunately, but this book, Günter Wagner's 2014 books, Homology, Genes, and Evolutionary Innovation, is absolutely brilliant. I highly recommend it. So he says the developmental mechanisms that produce novelties can be phenotypic plasticity and genetic accommodation. So when the plastic, environmentally induced change becomes genetically fixed. And the release of cryptic genetic variation. Again, we have to talk about robustness when we talk about network evolution. I think that's in about 3 modules from here. And then novel altered positional signals, okay, could trigger. So you suddenly have an organ that is in a completely different context. So it has completely different initial boundary conditions, so it will behave in completely different ways. And you could have the de novo origination of chins. How that works, we don't know, but they could be assembled by chance or arise through a duplication, which brings us to the underlying genetic mechanisms that generate novelty here. And examples that Wagner describes are so very important here, and we'll come back to that when we discuss disputes in evolutionary theory, cis-regulatory evolution is definitely extremely important here. So regulatory evolution. Transposition probably has an important role in the evolution of new cis-regulatory elements, and maybe more important here than in the adaptation of character traits. And gene-genome duplications, we'll talk about how they generate a sort of robustness that allows novelties and innovation to happen. So the basic claim here is that there is increasing evidence that there are different developmental and genetic mechanisms that produce adaptations versus these types of novelties. But one thing to notice here is that it's really hard to sort of delineate these novelties from adaptations. There is not sort of a discrete boundary. And this can be seen with the example of these modalities that are the same character identity with very different states. What makes it very different? You know, there's not a sort of a discrete threshold between a modality and just a variation in state. Okay, so very difficult to get a grasp on this, but some very good work done on the concept already of what is a novel trait. But do these theories really tackle this issue of evolutionary innovation and novelties? And think back to these sort of drastic novelties of new levels of organization. What's happening there? Of course, these theories are not designed to deal with that, but that's a completely different phenomenon, right? So these sort of theories have serious limitations, I think. And that shows you a lot about the status of theory in evolutionary developmental biology and evolutionary systems biology at the moment. Okay, I'm going to give you a Bible quote to motivate my final discussion here. Ecclesiastes 1:9 says, "What has been will be again. What has been done will be done again. There is nothing new under the sun." Whoever wrote this, supposedly King Solomon, was a true Newtonian, millennia before Newton. But so this is the main problem, okay? So if you look at variational structuralism, which is what Günther Wagner calls his theory, there's a bunch of obvious problems. After suffering through the first part of this course, you immediately notice it's too much focused on gene regulatory networks. It makes a few interesting points. So I do think that there is more to evolution than just adaptation, so that there are different mechanisms and different explanations required for novelty. I think both theories point that out very well, and especially epigenetic innovation theory, as opposed to Wagner's structuralism, points out that you need to consider more than just the genetic level to understand innovations. But it's formulated in a way that is very vague and doesn't take us much beyond the Vries and actually Darwin. So problems that have been formulated more than 100 years ago. We need to make some progress here. And we need to do this by looking at specific mechanisms and the precise meaning of the terms that are used in such theories. We'll come back to that problem. Okay, so for a theory to be useful, theory is extremely important in evolutionary biology, but for a theory to be useful, it can't be armchair philosophy. It can't just be throwing around some terms like emergence, self-organization, thresholds, okay? We need to define what these terms mean. Otherwise, we commit the same crime that Vries did when he said, oh, you know, the regulatory capacities of the organisms are explained by entelechy. This is just a word. It's not an explanation. Explanations, theories need to do work to provide explanations. So what is the work that these theories are doing here? Okay, I'm not quite sure. But the worst thing is that I think both of those theories miss a very important point about evolution. And that is that it is radically open in a way that none of these theories can capture. Okay, so the most important limitation of those theories is they're too narrow. Both of them are firmly stuck in Newton's mechanistic paradigm. They are doing science in a box. Okay, the configuration space of evolution is predefined, especially in epigenetic innovation theory, that takes dynamical systems theory to represent the developmental systems that produce the novelty. Okay, so these are well-defined systems with boundary conditions, et cetera, et cetera, et cetera, in a box with predefined rules. And as long as you have a system like that, as researchers in the field of artificial life have found out the hard way, you don't get true innovation in evolution. This is called the mysterious failure of a life. Okay, we don't produce living algorithms in the computer because we're missing some fundamental point. And what that point may be, it has something to do with a type of radical innovation will be the topic of next lecture. Thanks for listening today, and I hope to see you back next time. Bye now.